Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I'd like to invite you to mark your calendars for the very first Dr. GPCR Virtual Cafe held on March 26th, starting at 11 a.m. EST. Our first live virtual cafe guest is Dr. Brian Airy from Bristol Myers Squibb. Space is limited, so don't forget to get your tickets today. Visit drgpcr.com/virtual-cafe for more information. To present your original work or your company's technology, or to sponsor a Dr. GPCR Virtual Cafe edition, please fill out the form on the cafe's main webpage. The goal with the Virtual Cafe talks is to bring you GPCR-focused live presentations every month. You'll be able to join us live and interact with the speaker and attendees. We hope you'll join us. To stay on top of all your GPCR news, subscribe today to our monthly Dr. GPCR newsletter at drgpcr.com/newsletter. Last but not least, find us on YouTube. You can subscribe to our YouTube channel to watch the latest Dr. GPCR podcast episodes and much more. So hello everyone, this is Yamina from Dr. GPCR. Today we have a brand new episode of Dr. GPCR podcast, and I have the pleasure of talking to Dr. Ross Cheloha. He is a tenure-track investigator at NIH at NIDDK. Hi, Ross.
Hello.
It's so great to have you on the podcast. Thanks for taking the time.
Yeah, thanks so much for the invitation. I'm a big fan of the podcast, and I'm really happy to be able to chat with you today.
Super. I know that you're planning a move to the Bethesda area, so we're really grateful to have you with us today.
Yeah, thank you. It's a welcome break from packing.
That's awesome. That's awesome. So can you tell us a little bit more about your background and what were the different stages as in your life as a scientist that led you to your current position?
Yeah, definitely. So as far back as I can remember, I've been interested in thinking about diseases and thinking about ways in which you might be able to treat a disease. When I was young, I loved writing reports on different kinds of exotic diseases and what the symptoms were and how it was treated and what people are trying to do to treat it. So as I entered college, I had planned to become a pharmacist. That was sort of what I had thought I was going to do. But at that point, I sort of was taking lots of classes, and I became interested in some more basic scientific type research. And I decided to send out some emails to different lab heads at the University of Nebraska, and I ended up in the Department of Chemistry. And in the Department of Chemistry, I was thinking, I'm going to discover new drugs, and that's obviously not exactly what I did, but I did learn to do some chemistry using enzymes. I became really enamored with sort of this interface of chemistry and biology. And at that stage, I had to decide: do I want to go to pharmacy school or do I want to continue to do research? And at that point, I decided I really liked the research aspect of it. I really liked the freedom that came along with doing research, the ability to be creative, the ability to try to bring new approaches to tackle difficult problems. So I decided to attend graduate school at the University of Wisconsin in Madison, where I was in the lab with Sam Gelman, and I there learned to do peptide chemistry, which was really a valuable transition for me, because doing peptide chemistry, it's a fairly quick way to be able to make lots of different analogs of biologically active drugs. So that was really an exciting point for me in my development as a scientist, where I was able to actually make new molecules that had never been made before and see how they worked in cells and eventually in animals. So that was something that was really inspiring to me, and ultimately motivated me to continue on with this path of research. It inspired me to go do a postdoc here in Boston, where I am now. I did my postdoc in the lab of Ploegh at Harvard Med School and Boston Children's Hospital. And I learned some new techniques that really allowed me to develop sort of more distinct and new ways of looking at problems that are of relevance to different kinds of diseases. So I'm just finishing up my postdoc now, and I'm transitioning into my new independent position as the head of a lab at the National Institutes of Health, where I'll be able to lead different kinds of projects to hopefully bring new angles to understanding and studying disease.
That's fantastic. And it's funny because you said pharmacy, and I was sure you were going to say medicine. So how come pharmacy and not medicine?
I was always really interested in the drug angle of it. So coming
Okay.
up, trying new drugs, coming up with new drugs to treat problems. So I was always really interested in the types of things that scientists have come up with to try to reverse disease. And so obviously, physicians are very important in all these processes. I was never as interested in the patient treatment part of it. I was more interested in trying to come up with new things that were could be helpful in new ways. And so pharmacy was something that people in my family had done, and I kind of knew how that worked, working with drugs. But then no one in my family had ever done basic research before. And so I wasn't really familiar with that, but it proved to be just a really exciting way to be able to, try things that have never, ever been tried before. And that was something that was super appealing to me.
So more of a doer and more of a molecule synthesizer in a way, where you can
Right,
control what
exactly.
kind of molecules you can put together. So you mentioned that you just finished your postdoc and you transitioned as a lab head at NIH. Can you tell us a little bit more about that transition, and how come you decided to go this route and not, for example, in an industry position?
Yeah, definitely. Basically, in the past year or so, I've been applying for different kinds of jobs. So most of these have been academic type positions in IH. It's not exactly academic, but I applied for lots and lots of academic jobs. I applied for a few industry jobs. Um, Ultimately, I like the freedom of academics. So I have ideas that may not be directly relevant to producing a drug, but nonetheless, I think that they can provide new understanding that could lead to totally new angles. And so these aren't things that are going to turn into drugs overnight, but I think that developing new methods to study systems of biomedical importance, like GPCRs, it's really appealing to me, but it will take longer and it will take more freedom to be able to try to implement these things that no one has tried before. And the ability to really try um, try ideas that aren't directly translatable, immediately translatable to drugs, something that is more in line with academic or government research, and that's something that I'm really excited about. I have things that I think should be really interesting. It's not clear how they will apply to actually discovering a drug, but I think it's going to tell us something new about biology. And in the end, anything you can contribute to that end is hopefully going to provide a step forward in understanding disease and treating disease. So industry versus academia, I really gravitated towards the freedom of academia, the ability to try things that are not necessarily a new drug or a new way to treat a disease, but they are telling us something about biology. You can use creative approaches, and yeah, the freedom and creativity is what I was really attracted to.
You touched on a very interesting point when you said, industry versus academia. I think there is this misconception in academia that, industry is kind of the dark side. I think it's industry and academia are useful, are helpful, and are necessary.
Yep.
You need the basic science, you need the academic system so that you can be creative, like you mentioned. But then you also need that organization that industry has, and also the funding that industry has
Yep.
to take that discovery into the clinic, because ultimately everyone's goal is to improve human health.
Most definitely. And I I don't really, being in the type of position I'm currently in, I don't really have it in my head that I'm going to be able to treat a disease from this type of position. And I fully recognize and appreciate and value the unique role that industry plays, and it takes all different kinds of approaches to be able to push something from an idea to being able to try to treat someone. And it requires different types of approaches and creativity and resources along the way. And I think it's really pretty beautiful how the different types of players can all contribute to this really grand goal of trying to help people with disease.
Absolutely. So can you tell us a little bit more about the process of going from
from the thought of, oh, I want to be in academia because I like the freedom, to the point where you actually get an offer letter from an institute and says, welcome and do your work?
Yeah, yeah. I will say it's a very long process, and it's not straightforward. So the first thing you have to decide is, do you really want to do it? It's not something where you can fill out a one-page application and send in a CV and they say yes or no. There's a lot involved with it. You have to put together a research proposal on the type of work that you propose to do over the next approximately 5 years. You have to write statements about why you're a good fit for the institute, why you're a good candidate for promotion of scientific diversity, cultural diversity, ethnic diversity, whatever you can contribute to the diversity of the institute that you're going to. And then, so these places get hundreds of applicants, and most of them I never heard back from again. It's a little bit disconcerting, but it goes into a black hole and you basically never hear back. Or I'm still waiting to hear back. I applied about a year ago to a lot of these places and haven't heard back yet. But some of the places you will hear back from, and you'll go through different stages of the interview process, either a video chat type interview, or if you're selected, you go for an in-person interview where it's a 1 or 2 day long process where you present on the work that you've done and then present on the work that you're planning to do. The most exciting part is really just being able to take the work you're planning to do into a room full of people who know the basics of what you're talking about, or they should be able to understand the basics of what you're talking about, and then just trying to convince them that this is important work, that you are uniquely trained and able to do this kind of work. It's really exciting, but it's also exhausting. It was basically a full-time job for probably 6 months or so. I did a lot of travel before the pandemic broke out. I think I went on 6 or 7 interview trips, and from December until February. So it's really a lot of work, and it's something that you really have to be willing to dedicate yourself to give it a full run. It's not something where you can just say, hey, let's try this. It's really, I am grateful that I had a supportive environment where I could dedicate the time and energy and focus needed to be able to give this a fair go. It could be, I can imagine it'd be difficult in other types of circumstances where you have more constraints on your time, but I was able to put my full effort into this, and in the end, it turned out well for me. But for a long time, I didn't really know. I applied to, I think, 65 or 70 places, something like that. I got a handful of interviews, and a couple ended up in an offer, and I was really pleased with it. It also required supportive people on the back end. So I had people writing letters of recommendation for me, sending them to different universities, looking out for job postings that were a good fit for me. It took a lot of time and effort. It took a lot of support, including, professionally and personally. It was very stressful. But ultimately, it was really enriching, and it was super exciting to be able to just put ideas out there, get feedback on them, and say, this is what I want to do, and does anyone want to give me a place to do it? So it was really gratifying and exciting that there were a couple of places that thought it was the work I was proposing would fit well with what they were doing there. And yeah, in the end, it's a really exciting time, but it's completely wide open. So.
Yes, it's I can see the time commitment that's necessary for that. And I've never applied for an academic position, but I did apply for industry positions, and it's pretty much the same, in the sense that you do have to put the time and effort in order to show your skills, to show that you fit in the team, in the group, and the company culture, but also you have to put yourself out there. So it's a different type of preparation.
Mm-hmm.
You don't have to come up with a project for 5 years if you go to industry, but still, I think both ways of routes of going require time and effort and preparation.
Definitely.
And support, and a lot of support as well.
Yes.
So that's amazing. So let's shift gears a little bit towards the science. So how come you started working on GPCRs? What's the story behind that?
Right. So it all relates back to my PhD. So I mentioned I was in a lab where I learned to do peptide chemistry. The lab I was in really didn't have any special focus on GPCRs. They were more of a chemistry lab interested in making new kinds of peptides. And so we were looking for different types of projects that I could take on as a new PhD student. And we found what at the time was a completely receptor that I didn't know anything about. And it was a G protein coupled receptor, and it bound to a peptide hormone. So it's the parathyroid hormone receptor. It binds to a medium length peptide. Typically, I'm working with peptides that are about 35 amino acids. And it was really a great scaffold for me, sort of as a beginning chemist, to say, can I make different versions of this peptide? And so there are a lot of different versions of a peptide you can make fairly readily. And then the question was, well, okay, we can make these. What are we actually going to do with them? We can do sort of the biophysical measurements that chemists like to do, but that doesn't really tell us much about why we're making them. So we found a really great collaborator, Tom Gardella at Massachusetts General Hospital, who was an expert in parathyroid hormone receptor pharmacology, and he was very willing to test these peptides that I made. And for that, I'll forever be grateful, because I don't know what I would do in my current position if I got contacted by someone who said, "Hey, we've got a boatload of these peptides that we made. We don't know if they'll work at all. Will you test them for us?" He was very willing to take on this person he had never met before, test my peptides, and it was really great because I could make the peptide, send it to him, and then within a few weeks, he would be able to provide me with the biological data for cell-based assays to say, this one works well, this one doesn't work so well. It allows us to do lots of iterations. And so, yeah, that was really my thrust into G protein coupled receptor work, was this collaboration with Tom Gardella. And he's really a valued colleague and a collaborator, and someone who I continue to work with to this day.
That's fantastic. So I do know Tom, and Tom is amazing.
Yeah.
He is an encyclopedia of PTH receptor and peptides, and he is truly, truly a nice person.
Yes, indeed.
I'm going to say this here. I've been trying to get him on the podcast.
Oh, really?
To talk about PTH. So hopefully by the time the podcast airs, I'll be able to say I have Tom on the podcast. But
yeah, I'll push him towards that too. I'll reach out to him and let him know that I did this. And what a great experience it was. So
thank you. Yeah. So you're doing your PhD. You have all these peptides that bind PTH. I guess PTHR1. So there's two isoforms of the
right.
receptor, and then you get in touch with Tom Gardella at MGH. They test these peptides, and then what do you do next? What did you discover, and what do you do next?
Right. So the whole idea behind the analogs of peptides that I was making is that they contain non-natural amino acids. So the idea was that these would stabilize the peptides against breakdown by proteases. So this seems to be somewhat important in vivo in animals. And so the idea was that we would make analogs of this peptide that were stabilized, but could still serve to activate the receptor. So that was the basic idea going in. But along the way, we were able to do that. So we made lots of analogs, figured out where we could put the non-natural amino acids and retain good biological activity. But along the way, we sort of worked with Tom a bit, and we sort of took this side road where we were looking at the ability of these peptides to bind to the PTH receptor, either when it was coupled with G protein or not coupled with G protein. And so there's this whole subfield of PTH receptor work where they're looking at different receptor conformations. And so we found that putting in these non-natural amino acids could make these peptides that were selective for either one conformation or the other. So this was something I'd never heard of before. Being a chemist, I'm making peptides. And he says, "Do you want to try it in this assay?" I say, "Yeah, sure, go ahead. And, if you got the peptide, go ahead and try it." And so with that, we're able to make some peptides that showed some really interesting selectivity. And so that was nothing we had ever predicted would happen, but it was something that we sort of found along the way. So the take home, one of the big take home messages from that work with Tom Gardella is that we were able to make peptides that were stabilized against breakdown. They were still able to potently activate the receptor. And when you put them into mice, they induce more enduring responses. So PTH receptor causes a spike in calcium in the bloodstream. So these peptides induce really prolonged calcium spikes in the bloodstream. And so this was really gratifying because it was the first time we had seen that we were able to do this with this new kind of peptide that we were making. First time we were able to push it all the way to animal assays. But also, we also had this sort of new direction and using non-natural amino acids to make peptide ligands that are selective for different types of receptor conformation. So this has led to a whole lot of other different directions that continue to go on in the Gelman lab with respect to biased agonism and signaling and receptor subtype selectivity and all different kinds of selectivity from using these non-natural amino acids.
Wow. When you talk about these different peptides that stabilize different conformations and they're selective, can you tell us more about is there a different signaling outcome with these different
Right.
peptides?
Right. So the the thing that we've looked at the most with PTH receptor signaling is the duration of signaling in cell-based assays. So what
Okay.
we do is to apply these peptides to cells that express the receptor for some short amount of time, maybe 10 minutes or something like that. And then we wash them away. We remove the media, wash the cells, and then we say, how long do the cells continue to signal for after that? And
Okay.
so the cells that tend to bind to the G protein uncoupled receptor state tend to produce very prolonged signaling responses. And this occurs both in cell-based assays, but also in animal-based experiments. So this does seem to be physiologically relevant. And there are other ligands that don't bind so well to that receptor conformation. They produce short responses, both in cells and in animals. And this seems to be somewhat of a conserved theme from nature, because there are actually two different ligands that activate the type 1 parathyroid hormone receptor. One seems to bind better to this G protein uncoupled conformation than the other. And they do seem to have subtly different biology. So it might be we're relearning something that biology was already using, but we're relearning it kind of in a funny way by making these peptide analogs. But there does seem to be some sort of link to the biology that takes place in most mammals, just by sort of looking at the ability of these different types of ligands to bind to different receptor conformations.
Yes, I think I read a little bit about PTHR and then the different peptides, and I think if I understand correctly, the long acting, the peptides that act over a prolonged period of time, and I think cyclic EMP is the readout for this one, could be useful in treating I think osteoporosis.
Yeah, so
Or
it's very interesting, because some of these peptides that seem really strong and long acting, it's yet to be determined whether they'll be good for osteoporosis, because there's sort of this unknown cyclical aspect of activation of PTH receptor that's needed to treat osteoporosis. So if you infuse PTH continuously over time, you will see the typical response. But when you get to the level of bone buildup and bone breakdown, which is what you care about in osteoporosis, continuous infusion actually causes bone breakdown. What you need is a spike in response, and then for it to diminish over time, and then you spike again. So it needs to be a cyclical activation. So whereas the long-acting PTH receptor might induce very prolonged responses, there's some concern that it might not be the best for treating osteoporosis. However, there are patients that lack their own parathyroid hormone. So they get the parathyroid glands removed during surgery, or they have some defect in biosynthesis. And for those patients, having a hormone replacement therapy could be really valuable, because the lack of parathyroid hormone actually causes a lot of defects in calcium homeostasis. So in cases like that, the long-acting parathyroid hormone might be beneficial, or more beneficial.
I read somewhere that actually the PTH function also regulates the teeth coming out. So the
Yeah,
adult teeth, which I find it fascinating that
right.
you're regulating that. Um, so you finished your PhD, and then what motivated you to do a postdoc, and did you continue working on GPCRs?
Right. Yeah. So it's sort of a circuitous path to my continued work on GPCR. So at the end of my PhD, I decided I wanted to learn more about immunology. That was about the time that the checkpoint blockade was really making waves. Patients with melanoma who were on death's door were basically being revived by these immunotherapies. And so I was really inspired by that. And I thought, well, I want to learn some more about immunology. I learned a little bit during my PhD. So I looked for labs that did immunology and chemistry. And there are actually not that many labs like that. So I found the Ploegh lab, hit up Plough's lab here in Boston. At the time, he was at MIT before he moved. And I said, hey, can I come be a postdoc for you? I want to learn immunology. And he said, sure, that sounds great. I was able to join his lab, get a fellowship to support my work for a few years. One aspect of his work that was sort of just coming of age when I joined the lab was his use of single domain antibodies, or nanobodies, which are an interesting byproduct of the immune system of camels, alpacas, and llamas. which have a number of practical advantages over conventional antibodies. And so in his lab, I learned to make these nanobodies, express them in bacteria. I learned to functionalize them with chemical modifications. And having learned these skills, I circle back to something that I've always been interested in, the ability to target G protein coupled receptors. And so using these nanobodies combined with chemical modification methods, I was able to link nanobodies to ligands for G protein coupled receptors. And at the beginning, it was really just a question of, can we do this? But as we figured out that it did in fact work, we were able to make some conjugates of these nanobodies and GPCR ligands that had some really useful properties and in ways that we didn't necessarily expect at the outset. And it's really a wide open field. And I think that these tools that we make should be useful to inform us on how GPCRs function, basically as mechanistic probes to understand how different facets of GPCR signaling affects the outcomes that we ultimately see.
Wow. And did you focus on a specific family of GPCRs during your postdoc, or?
Yeah. So I mentioned my continued collaboration with Tom Gardella. We actually went back to the PTH receptor. It's a favorite of mine. There's a rich set of mechanistic questions that you can look at with GPCR signal or with PTH receptor signaling. So being in Boston next door to Tom, I said, "Hey Tom, I've got an idea. Can we try some stuff with PTH receptor?" And he has so many tools that it was really a big boost to being able to ask some really pretty tricky mechanistic questions just based on the vast library of different types of receptor constructs and ligands that he had already made. So with that, we were able to sort of get quickly into asking what are really pretty tricky questions. And we were able to show that the ability of a nanobody to bind to a receptor can turn what's normally an unselective ligand into one that's selective for the receptor that's bound by the nanobody. So that was really probably the most exciting thing to come from this study, is that we're able to turn what are pretty crappy and not very selective ligands into ligands that are both fairly potent and selective, but also turn that ligand that didn't show any activity in a mouse into one that does show activity, just because of the boost that you get from connecting it to this nanobody that binds to the receptor of interest.
Was it because the nanobodies stabilize the peptide?
It's still sort of an unknown area, but the ligands that we used were sort of what I would call suboptimal. They were ligands that didn't themselves bind very tightly, whereas our nanobodies bound pretty tightly. So you can imagine the nanobodies sort of giving these moderately active peptides a boost just by sort of, I imagine them being anchored to the receptor. You have the nanobody, then the ligand can just sort of activate the receptor after the nanobody anchors it into place. That's kind of what we think is happening, but there's some interesting biophysical aspects of it that aren't entirely clear, but that's sort of how we imagine this working. Whether that's true or not, I don't have any proof of that, but that's sort of what we think.
Okay, and were these peptides the quote unquote normal length of PTH hormone, about 34 amino acids?
Yeah, so these peptides were shorter. They were 11 amino acids. They are from this series of peptides that Tom Gardella had reported a decade or more ago called modified PTH or MPTH. And so they were peptides that bound fairly weakly, but yet they were fully efficacious. So they just didn't bind as well. So we're basically able to restore the binding ability of these peptides with the addition of a nanobody that bound tightly to the receptor.
Wow. And what was the effect on the efficacy of these peptides?
Yeah. So in terms of cell-based assays, the peptide and the nanobody peptide conjugates were both fully efficacious, but the difference in potency was sometimes up to 10,000-fold, 4 orders of magnitude more potent when you use this nanobody to target these ligands to their site of action. So we can get huge boosts in potency through sort of our engineered binding.
That's phenomenal, because if I remember correctly, this modified PTH 1 to 11, this 11 amino acid short peptide, was already pretty potent in the nanomolar range, I want to say.
Right. Yep. So there are lots of different fine tuning things. The one we were using was about 100 nanomolar. So we could improve it down to sub nanomolar potency in some cases. And I think one of the other really exciting things is that we were able to use engineered versions of the receptor that basically lack the extracellular domain, where you replace the extracellular domain with yellow fluorescent protein in our case. And for those receptors, there aren't really any good ligands for activating super potently, because the natural peptide doesn't work very well anymore because the receptor lacks the extracellular domain. But if you instead use a nanobody that binds to YFP, it's like you have sort of a pseudo extracellular domain. You can have a binding event that doesn't happen with the natural peptide, but does happen when you have the nanobody. So on that type of receptor, we can have a conjugate that's a sub-nanomolar potency, where the natural peptide is at a level of something like a couple hundred nanomolar. So we're looking at a several thousand-fold improvement relative to even PTH 1 to 34 in these designed receptors.
Interesting. So looking at the big picture, what's the use of having these nanobodies?
Yeah. So I think probably the most valuable practical aspect is this one of selectivity, as I mentioned before. So there are two subtypes of PTH receptor, as you alluded to earlier. They're both activated quite potently by PTH 1 to 34. So there are ways to change the structure of PTH 1 to 34 to make it more selective. But what we showed is that if you take this fragment, PTH 1 to 11, and you attach it to a nanobody that binds to one subtype of the receptor, but not the other, what comes out the other end is a very selective ligand, especially when you consider that PTH 1 to 34 itself is not selective. So you're able to engineer in selectivity by virtue of the specificity of your nanobody. And so that's really valuable, I think, because antibodies and nanobodies are really the gold standard of what people use to selectively detect and target different types of proteins. If you say, is this protein present or not, you say, well, is there a good antibody for it? And so people are always trying to make good antibodies for their targets, sometimes with varying degrees of success, but people are definitely trying. And so the ability to use the selectivity of an antibody, or in this case, nanobody, to be able to selectively target a receptor, I think provides a really nice avenue for hopefully being able to engineer in specificity for your receptor of interest with just using the selectivity that's been identified with existing antibodies or nanobodies.
It's an interesting tool to have, and it's an important tool to have as well, because as you mentioned, there are very few antibodies that selectively detect membrane proteins or GPCRs.
Right.
You may or may not get lucky if you can purchase it.
Right. Yep.
And sometimes in tissues, one thought I had while you were mentioning the selectivity. So I worked a lot on chemokine receptors, and I became fascinated about the word escapes me. So this chemokine receptor CXCR3 has different splice variants.
Yep.
And so the canonical receptor is called CXCR3A, but then CXCR3B has actually a longer N-terminal.
Okay, yep.
N-terminus, and then the alternative, which is CXCR3 alt, is reported to be a much shorter receptor and apparently would have only 5 transmembrane domains.
Okay.
And there are a bunch of studies that came out that showed that depending in various cancers, I think it was prostate cancer, you have a difference in expression of these variants.
Yep.
But then you cannot differentiate between them.
Right. Yep.
Having a tool like this to be able to use as a marker, for example, and look at tissues and be able to say, well, it's this isoform in this disease and it's not that's priceless.
Yeah, I think that's a really valuable set of tools and a good goal to try to be able to produce antibodies to target these different receptors, or antibody conjugates that can target these different receptors. I think that could be enormously valuable for lots of different kinds of things.
Absolutely. And then would these nanobody coupled or conjugated peptides be useful in a clinical setting?
Right. So as we've kind of discussed before, PTH 1 to 34 itself is already quite active on the wild type receptor. And despite the fact that it seems to target both type 1 and type 2 receptors quite well, there don't seem to be too many off-target side effects. But what I will say is that So the main side effect is high calcium in the bloodstream. That's not actually desirable when you're trying to induce bone growth. That's sort of a dose limiting side effect. And so one thing that I think could be really exciting is that if you can use a nanobody or an antibody to target I would say, promiscuous ligand to a specific tissue, that could be a way to get tissue-specific activation of a receptor where you can promote the desired physiological outcome while avoiding the tissues where you get the side effect outcome. So for PTH, the bone is where you want to activate. There's also a receptor in the kidney, which tends to produce a lot of hypercalcemia. So I think that this ability to connect ligands to antibodies presents a really potentially useful way of promoting sort of desired therapeutic outcomes while avoiding side effects. That remains to be seen whether that will actually be true, but I think there's some hints that this could be a really valuable way of improving sort of the side effect profiles of GPCR ligands.
Yeah, and in any case, right now with these are valuable tools to at least dissect the different functionalities of, for example, the two types of PTH receptors.
Exactly. Yep.
And that's also very important to study and to know, because if you want to target any PTH receptor in the context of osteoporosis, but there's hypoparathyroidism, which also a mouthful,
Yes.
then you really want to make sure that you're targeting the right receptor at the right place.
Definitely. Exactly right.
So then you continued working on PTH receptors during your postdoc. And then what's next? What are you going to be working on as an independent investigator?
Yeah. So I'm really excited to ask mechanistic questions of G protein coupled receptor signaling using the same kind of tool. So the PTH receptor engages in a number of different types of signaling processes that have recently come to light. So the two that I'm, I guess, most interested in are endosomal signaling. Some peptides seem to be able to continue to signal in the endosome, whereas others don't. And there's not necessarily a clear rationale for why some ligands do this and some don't. And then the other signaling thing that I'm interested in is the duration of signaling. So there's long been this thought that tighter binding ligands will signal for longer. But with the emergence of this endosomal signaling, I'm not convinced that that's necessarily the case. I don't think that that's necessarily the whole story. So what I'd like to be able to do is to really develop mechanistic tools to say, how does binding dictate endosomal signaling, duration of signaling? We're going to look at biased agonism. We're going to look at the other types of responses for GPCRs. And so we'll have in our hands some what I think are fairly unique mechanistic tools to be able to tune different aspects of our ligand, because we really have a ligand that's been split into two pieces. And so we can tune each piece to the ligand independently of the other. And this will allow us to get down to some really what are tricky mechanistic questions to ask with conventional ligands, which is how does affinity affect these different processes? And it's really only using these ligands where they're split into the two different pieces can we really drill down into asking how does part A and part B contribute to what we're seeing in the context of a cell or an animal. And I think that should be really valuable to guide the future design of ligands for GPCRs with this information in mind.
I think it's valuable, and it's something that for a very long time we were unable to capture, is what is the effect on the cell, for example, of the length of that signal.
Yep, right, exactly.
We've been doing experiments, and you stimulate for 5 to 10 minutes, and then you measure endpoint, and you have something, but that doesn't mean that it stops within the cell, and maybe we're losing all that information.
Yep, definitely. And I think for PTH receptor, it's apparently important for PTH receptor. I would be willing to guess it's important for lots of different receptors, but I think in large part it's been missed. So.
Yes. And now we have the tools to do it. And are you going to focus on PTH receptors, or are you going to widen the net a little bit?
Yeah. So I think PTH receptor is a good place to start. I also have designs on moving to other kinds of receptors. It's funny you should bring up the chemokine receptors. Those have peptide ligands. I'm also interested in those. Yeah. I'm also interested in receptors where the they have small molecule ligands that are fairly synthetically tractable. The adenosine receptors are another one that is really of interest to me. I'm also interested in, I'm not, I'm willing to work with lots of different types of receptors. I hope that this technology will be broadly applicable. So I have these ideas about places to start, but I'm really always looking to try to move this into as many different types of systems as I can get into, because different receptors have different quirks and different types of features. And I think that by pushing this technology as far as we can, we'll hopefully be able to reveal some interesting biology in different contexts.
I'm sure you will, and NIH is a very good place to be doing that. You were explaining the type of receptors, and I already could think about people who were working on some of these at NIH, and I think you're going to find that you'll get the support that you need to test out your ideas. So we've talked about GPCRs and drug targets. One of my questions that I always send out, and I ask my guests, is, do you think GPCRs are a good drug target? What are your thoughts on it?
Yeah, so short answer, I think that they definitely are. I think that we're beginning to sort of realize all of the different parameters that targeting a GPCR can control. So I think, in the past decade, biased agonism has really come to the forefront. A lot of people are really coming up with unique ways to look at biased agonism, see whether that translates into unique therapeutic profiles in vivo. I think there was recently an approval of a drug like that. It's a really exciting time for biased agonism. I think that there are other aspects of GPCR signaling that are ripe to be pursued in the same kind of way. So duration of signaling is another one where I think it's really important. Endosomal signaling, I think, could be a really valuable one to look at. And then, of course, there are ways of targeting GPCRs in different tissues, different types of heterodimers, homodimers, whatever. So I think that the biology of G protein receptor signaling is continuing to be realized for how expansive that it is. And I think as we can begin to control these types of things, I think we're going to get new sort of ways to approach disease that we didn't even realize we could try before. So I think that I think there'll continue to be sort of traditional methods for looking at GPCRs that I think will yield useful results. But as we begin to really be able to finely tune the types of GPCR switches that we turn on, off, or toggle or switch to the side, I think that we're going to continue to get new kinds of tools and hopefully drugs that will allow us to treat disease in new ways.
I think it's a good time to be working on GPCRs.
Yes.
We do have a monthly newsletter, and I am amazed at the number of papers that come out every month in the various areas of GPCR research. I think there were 60 papers in the past month that came out, which is phenomenal when you think about it. And then so now that, although this episode will air in January 2021, we're in September 2020, and in this month's newsletter, I think we listed at least 4 or 5 GPCR related conferences and courses coming up in the next 2 months. So in spite of the pandemic, in spite of the fact that everything has changed since February or March, there is a strong community around GPCRs, and people are actually getting those papers out, and that knowledge the knowledge base is getting bigger and bigger.
Definitely, it's a super exciting time.
I think so too. So what would be your advice for junior scientists who would want to contribute to the field?
Yeah. So I'll start out with a disclaimer. Hopefully this is not too discouraging, but science is hard. I don't think that's really a surprise to too many people listening in, but science is not for the faint of heart, and it's not something that you're gonna want to do if you don't care about it. So my advice would be find something you really care about, that excites you, that interests you. For whatever reason, you may think that the science or biology, pharmacology aspect of it is cool. Maybe you have a personal connection. Maybe you want to help a lot of people. It doesn't matter. Find something that really excites you, and then devote everything you can to trying to contribute to the field. Read papers, attend seminars, look at TED Talks if it's something you don't know anything about, read Wikipedia articles, give everything you have to trying to get into an area that really interests you. Because if you can get into something that really interests you, it's not really, so much like work anymore. It's something where, you can really be excited and interested and engaged in what you're doing on a daily basis. And for me, that's been really great. I've loved thinking about making molecules to treat disease for as long as I can remember. And, I've just been continuing to be able to do that. And it's been really a treat for me to just be able to work every day on things that excite me. And I wish, for whoever is listening to this podcast, that if you're considering going into GPCR work or scientific work more generally, find something that interests you and excites you, because it's really a rewarding thing to do. Throw everything you have at it. Leverage your networks. Just do whatever you can to get into an area that is of interest.
I could not agree more. I remember the first time when I heard about GPCRs, it was in an undergraduate class, and I told myself, "Wow, this is really cool." And I worked, so since I started undergrad up until this year, I actually worked in the lab, and I enjoyed every moment of it, and the satisfaction that comes out of it. Well, that one time when that experiment finally works and it's 10 p.m. and you cannot share that enthusiasm with anyone because already everybody left. That makes up for all the times that, your cells died, or your experiment didn't work, or you mixed something, or you accidentally threw out the samples that you actually needed. But yeah, science is hard. I think you absolutely have to love what you're doing. You have to make sure that you choose a good team to be part of. Great mentors. You touched on it in the beginning, is that you had the support that you needed in your postdoc to go and take the time to plan out the next steps in your career, and also the support you needed to do these experiments and to be successful as a scientist. I think that's also something that needs to be done. And I really liked when we were talking about you choosing a postdoc, the fact that you just sent an email to your professor and said, I like your work, can I come?
Yep.
And for graduate students and for students out there who are listening to the podcast, it is that simple. Read papers, get in touch with people, send an email, get on the phone. Based on my conversations and on my experience, just send an email. And most of the time people will answer. Maybe they'll say, hey, I don't have the space, or I cannot have you on. But then again, you create that connection.
Exactly. Yep.
And you never know. Yeah. Super. So one of the questions that everyone seems to like about the podcast is, at the end, is what are the top 3 aha moments that you had as a scientist during your trajectory?
Yeah. So my trajectory as a scientist has been kind of winding. So the specific details of these experiments have sort of been all mushed together in my head. But I have 3 different sorts of experiences that have really left a mark on my scientific thinking. So the first one was when I was an undergraduate researcher. So I joined a chemistry research lab when I was a freshman. And it was an organic chemistry research lab. And I hadn't taken organic chemistry. I had never done lab work before. Scientifically, I was a liability to have there. I didn't really know what I was doing. I was assigned a postdoc to work with and they were really nice and helpful. They would sort of set me up, and I would help them a little bit. They would help me a lot. And I wasn't very good at the beginning. I didn't really know what I was doing. I was basically running the control experiments the entire time I was there and would never do anything new. But after a while, I eventually was able to run an experiment where the outcome of the experiment was unknown and it was new data. And in my case, it was using an enzyme to run a reaction where we didn't know if the enzyme would catalyze the reaction or not. We didn't know what the product would look like. I was able to show that this enzyme catalyzed a reaction and gave a good result as far as we were concerned. And that was really, it was so exciting. It was a very simple thing, and it didn't change the course of any project or anything like that. But just being able to produce a new piece of scientific data was something that was super exciting to me and really gratifying, really powered me through all the things I messed up over and over. So that's number one, producing a new piece of data, no matter how how small was really valuable. The second one was, I think I might have alluded to this earlier, was when I had made the PTH ligands and I had sent them to Tom Gardella. So this wasn't as much of an in-lab aha moment as it was, oh, look, it's an email from Tom, and he had sent the preliminary data from the first set of peptides I had sent him. And it was I think there were 3 different types of assays he had run with like 6 different peptides. And I was just mesmerized by it. I looked over it over and over. I'm like, oh, wow, what could that mean? What could this other thing mean? What kind of peptide should we make next? What does this tell us about the receptor? Just being able to have data and analyze data based on completely new things, and thinking about what that means about biology and pharmacology and drug design. That was my second really aha moment, where it was just like, oh, this is so cool. I want to keep doing this. Like, this is such a exciting, but also like propelling experience. Like, I want to do more of that. Third one is an experience where I just designed something just because I could, where I didn't necessarily have an underlying scientific rationale for trying this. I wasn't thinking this is going to be a new drug. I just thought of something. I said, hey, I wonder if this would work. And so this relates to the nanobody-PTH conjugates. And the very first experiment where I was able to finally make the conjugate of the nanobody and PTH, which was, after I figured out how to do it, it was fairly trivial. But at the beginning, I was having problems. And so I was eventually able to make this. I was able to express the receptor that had the right tag incorporated into it. And everything worked. The negative controls worked like they're supposed to. The positive controls worked like they're supposed to. And I was convinced that I designed something that worked, where it was not at all obvious that it was going to work. So with that, I was just so excited, and it opened up so many doors. And it was just really a moment where I thought, wow, I can design new things that will actually open a lot of new doors to looking at GPCR signaling and cell biology more generally. So yeah, the third aha moment was just having this experiment work with all the positive controls and negative controls was something that was completely new, and there was no reason that it necessarily had to work. It could have failed completely for a lot of different reasons, but it didn't, and that was just so exciting for me.
That's awesome. And that I feel like that the third aha moment that you just mentioned, I think it comes out of experience.
Right.
It's that it comes back to the first aha moment where you actually were able to do something on your own and you produced data for the first time, but then you built on that momentum and you got that experience. And I feel like maybe it's just me, but at some point when I got very used to doing some things, I had this intuition saying, well, let's just try this way because I have a feeling it will work.
Right.
And you still have to design it, but that comes from experience. That doesn't come, that comes as a result of so many failures.
Right. Yep. Definitely.
Which is phenomenal. And I think it's also fun. Your second aha moment that you mentioned, getting the data back from Tom Gardella, and then feeling that you actually made something useful that actually is active. Yep. It's just phenomenal. It's just a great feeling. From the perspective of a molecular pharmacologist, I love dose response curves. So I can have tears in my eyes seeing those, nice EC50s. The fit is great and it's actually active. It's just priceless.
Definitely.
Super. So last question. Now you're opening your lab. You're going to have to build your own team. Do you have any job openings, and where can people find you?
Yes. I have one postdoc job opening. I have an ad posted on Job Archive. I think if you search for my name on Job Archive, it'll come up. I think I have a posting up on the Dr. GPCR website as well. So yeah, you can find the posting either of those places. One ad I don't have up yet, but I will be looking for is a post-bac researcher. So this is someone who has completed their undergraduate studies and is looking to get some research experience after undergrad to decide what they want to do next. So that will be on the NIH website. There's a very specific mechanism for applying for that. And once I actually start my job, that should be up. So yeah, would love to hear from many of you.
Super. We will. I know that you sent in that postdoc job ad. It's on the website. People can find it. But then again, I think people just can find you on Twitter or can find you on social media and on LinkedIn and soon at NIH, and just reach out to you.
Definitely.
Super. Thank you so much, Ross, for your time. I really enjoyed talking to you, learning more about PTHR and about nanobodies.
Yes.
We wish you the best of luck with the move, first of
Thank
all,
you.
and then best of luck with your research. And hopefully we can check back with you in a couple of months and see how it went. And then you'll tell us more about your new lab and your new team.
Definitely. That sounds great. Thanks so much for having me today, Yamina.
Super. Thank you so much, Ross.
Yeah.
Bye.
Thank you for listening to this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Don't forget to subscribe to the Dr. GPCR newsletter at drgpcr.com/newsletter. Until next time, stay safe.